So like I had mentioned earlier, please understand that these are totally supplemental material for the course. This is not a substitution for this. I'm not going to be able to provide any nice pictures. If you're enrolled in this class right now, your textbook has amazing pictures. I'm just providing you concept maps and kind of talking along the way as I'm doing these. So for the active sites of enzymes and then the actual reactions themselves, this really comes down to the ES complex, the enzyme substrate complex. The other thing that I wanted to mention in this video would be the two hypotheses of how things work, or really just the two things that explain this. You should consider them reaction hypotheses. There's two of them. There's the induced fit and then the lock and key. So lock and key, and then there's the induced fit, which that should be, this is all review, right? So lock and key just says that the enzyme and substrate fit to each other with the like a lock goes into a key, that they're that specific. I'm just going to go ahead and say this. The lock and key makes a lot of sense pedagogically in terms of explaining the specificity of enzymes and the specificity of their substrates. And it's a really good way to explain children in 6th grade how enzymes work. That's not how enzymes actually function. What is a more valid explanation of really how they work would be the induced fit. And what this is to say you have your enzyme, this is the active site here, right here, and then here's your substrate for it. Right here, right there, okay? So as they start to bind here, and this is binding is happening, oh, this is really getting frustrating. As they start to bind here with the active site, and keep in mind that these are non-covalent forces here. Non-covalent forces here. So as they start to do this, this is going to cause a conformational change, which is going to cause the breaking and reforming of the covalent bonds in the enzyme's three-dimensional structure, causing it to ultimately change shape. And as it changes shape, the active site changes, and that becomes more appropriate to bind to that. My guess is that if you think about how an enzyme works, that having an enzyme active site that is conducive for this reaction is arguably not the most thermodynamically stable position for them to be. They're not going to go into that position unless they have something that's going to give them energy to do so, which we'll talk about what that means later. But anyways, the induced fit is basically just saying that enzyme, the ES complex formation is by conformational changes. From a conformational change. That's all that we're talking about with that. Now, let's move on to the ES complex. So for the ES complex, the things that I wanted to mention would be the active sites themselves and then the binding energy associated with these. And this is a little weird to mention, but active sites, as I think I've talked about, or probably you had your general biology professor talked about this, is that this is a conducive environment for catalysis. What that means is this is the appropriate conditions at which we need to have this reaction proceeding at a much faster pace. Think about it as reactions with palladium on carbon we talked about for organic chemistry versus like that of lithium aluminum hydride. But anyways, so this is a conducive environment for the catalysis. The active sites tend to be, I'll just go ahead and include it up here, they have two parts of them. So there's a hydrophobic base. Hydrophobic base, so if we were to draw our enzyme active site here, whoop-de-doo, that's our enzyme. At the base here, this area that I'm shaking with my pen here, this is all hydrophobic, but at the tips of it, at the very tips, so hydrophobic base, it has a hydrophilic hydrophilic tips, okay? So the analogy that my professor gave us was that it was like a church. So if you go to a church, there is a steeple on top of the church that's at the very high part of it, but at the base of it, everything is kind of here. So here's the hydrophobic base of it, and at the top of it is where the hydrophilic tips are. Active sites are also where cofactors can bind to give us the apoenzyme versus the holoenzyme. Apoenzyme being something that doesn't have the cofactor, holoenzyme being something that is whole. I'm not going to stress that. That's a terminology thing. You can figure that out later. Anyways, so the formation of the ES complex, the actual formation where we take the enzyme and it reacts with the substrate and reacts in equilibrium to form the ES here. Let's talk about what this is. So this the binding here, like we said, how it causes a conformational change. Well, where does that energy come from, that conformational change? That comes from something known as binding energy, which I'll talk about later. But this facilitates the formation of the transition state. This facilitates the transition state formation. This formation here of the enzyme substrate conflict, like I wrote up here, it's non-covalent factors here. This is not a covalent bond. This is intermolecular forces for the most part. And then the next thing that I'm going to talk about, and I'll do it in a bright blue, is the concept of binding energy, which drives home a lot of things. This is important for not just for this class, but for understanding biology as a whole and understanding any chemistry that's really worth doing. So over here we talked about how this bond forms here, this undergoes a conformational change, and that conformational change creates an inducive environment for the enzyme to catalyze the reaction. This is not the most stable thing in the world. It's stable, yes, in that it exists, but it's not the most stable conformation in the world. And there's this balancing act with binding energy that we have of how much energy we're going to get to overcome that hump. Because even if you remember back to when we talked about the Gibbs free energy thing, that even a spontaneous reaction, I'll draw it. I'll just go ahead and draw it. Even a spontaneous reaction where the reactants are in a higher energy state than the products, which this is, even that has an energy curve that it must overcome. So the binding energy is just that. It's the energy that we use to overcome that hump. So binding energy is the delta G that's released upon substrate binding, which is why it's called that. Free energy released binding. Okay. So what is this free energy going to be used for? Well, in the case of other things, it can be used for a lot of things. But in this, for enzymes, it is used to lower the activation energy. We lower the activation energy, we have facilitated the formation of the transition state. Make sense? Hopefully. So let's talk about what this means if it's too high or if it's too strong. If it's too strong or if it's too weak. And it is a balancing act. So if it's too strong, let's talk about what happened if the binding energy were too strong. If the binding energy were too strong, we would have a lot of energy going into our reaction. And if we have a lot of energy going into our reaction, that energy is going to be used to convert a bond, right? So if I put a lot of energy into a reaction, I'm going to put a lot of energy into the formation of a bond. I'm going to have a very energetic, a very strong bond. So if it's too strong, say for example this were to form a covalent bond, then the transition state would be too stable, and if the transition state was too stable, what would we have? Well, if the transition state was too stable, we would have ourselves a permanent inhibitor, or a poison, if you could think about it. And that's exactly what we do. So say I wanted to make a compound that would kill you. Okay, I'll say cyanide, right? So if I'm a Cold War pilot and I get shot down over Korea, I'm going to swallow a cyanide capsule so that they can't interrogate me. Cyanide works just this way. It forms an extremely strong covalent bond with the enzymes of the electron transport chain. And by doing that, blocks all production of ATP. Another example would be ricin. Ricin inhibits your ribosomes, all right? So it forms a permanent covalent bond to the RNA. Clindamycin, if you've ever taken that, that's a covalent bond is being formed in the ribosomal RNA of bacteria when you do this. So covalent bonds, yes, very strong. Poisons, lowers the EA. I just want to make one last thing here, that poison, or you can think of it as permanent inhibitors, these are very similar to the TS. The transition state is very, very stable. But if it was too weak, by the same notion, if it was too weak, we would have no energy, or not enough energy, to pass the activation barrier / stabilizing the formation of a transition.